Welcome on this webinar devoted to a new molecule called Crystallophore number 1. In this presentation, we'll present the unique properties of Crystallophore number 1 for protein structure determination so that it will become your essential partner in your crystallographic work. Crystallophore is a lanthanide complex that will both facilitate the crystallization step and the structure determination process, thus solving the two major issues of macromolecular crystallography. To start with, Crystallophore is a new additive acting as an efficient nucleating agent. When added to the protein solution, it provides new crystallization conditions and improves crystals quality. Crystallophore is really easy to use. Just add 100 microliters of your protein solution into the tube as provided by molecular dimensions, mix, and centrifuge it to get a solution ready for conventional crystallization screening. To demonstrate the nucleating properties of Crystallophore, we use a set of 8 proteins, including commercial proteins, proteins produced within our lab, as well as 2 proteins with unknown structures. Our approach was quite simple, in the sense that for the 8 proteins of our panel, we have compared the crystallization results obtained for the protein alone and for the protein in presence of 10 millimolar Crystallophore. 576 crystallization conditions were systematically screened, including different commercial kits. The details can be found in our paper. Here are the results of our comparative experiments. We have represented the number of conditions leading to crystals for each protein. The hits for the protein without Crystallophore are represented in gray, and the ones obtained with Crystallophore with dots. Consequently, this histogram provides information on the number of hits obtained for the native protein only, on the number of hits that are in common, and you can already see that some hits are unique to the presence of Crystallophore. First, 15 to 100% of the native hits are reproduced with Crystallophore. Second, as already mentioned, Crystallophore provides 10 to 85% of new unique crystallization conditions. And you can see that this is also true for the 2 proteins with unknown structure, for which in the presence of Crystallophore led to 30 to 50% of new crystallization conditions. Here are some examples of crystals obtained in the presence of Crystallophore to be compared with the equivalent conditions for the protein in the absence of the additive. You can see that the crystals produced are ready for X-ray diffraction experiments without any optimization. To go further in the properties of Crystallophore, we have determined crystallization diagrams for different proteins of the panel. Here are the diagrams obtained for one of the 2 proteins with unknown structure, PB6. The black dots indicate drops with crystals. You can thus see that the addition of Crystallophore during the crystallization process induces an important enlargement of the nucleation zone. The first consequence is that Crystallophore will facilitate the detection of crystallization hits. The second important conclusion is that crystals can be obtained at low protein concentration, as crystal can be grown at concentration of 5 micrograms per milliliter in the case of PB6. This can be a real advantage for precious samples or difficult to produce ones. Under UV light, Crystallophore emits a green light that can be useful to detect crystals within drops and to discriminate between protein crystals and salt crystals, for example. Finally, our observations are not restricted to our test panel. Over the last 18 months, we have collaborated with different teams leading to confirm our initial findings that Crystallophore provides new crystal forms and that, in most cases, the produced crystals are ready for diffraction, as you can see on the different photos. Crystallophore can also be used as a phasing agent to facilitate the structure determination process. Indeed, the molecule contains a terbium atom that presents a huge anomalous signal 3 times larger than the one of a selenium atom. This anomalous contribution remains important even on an X-ray home source. Our protocol for phasing is as simple as for the crystallization protocol. We have checked that Crystallophore is compatible with most of the cryoprotectants conventionally used. So, our protocol to prepare derivative crystals with Crystallophore consists in adding 10 microliter of your cryoprotectant solution into the tube as provided by molecular dimensions, and in soaking crystal during the cryoprotection step for only a few minutes. Using our phasing protocol, we have been able to solve de novo all the proteins of our test panel. This includes, of course, the 2 proteins of unknown structure. In all cases, the high quality of the phasing process allowed to automatically build a large portion of the atomic model. Over our collaborations, several proteins have been determined. The quality of the experimental electron density map obtained thanks to phasing with Crystallophore highly facilitates automatic model building. This second example also illustrates the high phasing power of Crystallophore. Of course, both nucleating and phasing properties of Crystallophore can be exploited, as in this first example where 2 new crystal forms were obtained and exploited to determine the structure. This last example illustrates the gain in time Crystallophore can provide. In that particular case, the native crystals only showed diffraction at around 3 angstrom, with presence of twinning avoiding structure determination. As for the previous case, both properties of Crystallophore were exploited. The new crystal form provided by the additive presented no twinning and resolution up to 1.7 angstrom resolution. The structure was easily solved thanks to the anomalous properties of Crystallophore. To conclude, Crystallophore is a unique tool that can be used as a nucleating agent to solve your crystallization issues and may facilitate crystal detection. It is also a powerful phasing agent that may highly facilitate your structure determination. And of course, as illustrated during this webinar, both properties of Crystallophore can be combined to increase the efficiency of your crystallographic work. We finish this presentation by mentioning that, to our knowledge, and over the last 2 years, about 20 structures of proteins have been determined thanks to Crystallophore, including our test panel, but also 2 unknown protein complexes.